Okay, so the last speaker this afternoon before the round table is Dr. Sachdev Sidhu from the University of Toronto. Dr. Sidhu trained at Simon Fraser University and subsequently at Genentech before being recruited to the University of Toronto 5 years ago. Dr. Sidhu focuses on synthetic antibodies and will talk this afternoon about synthetic proteins and their use as new tools for new biology.
Thank you very much. As I would like to once again congratulate Greg Winter for a fantastic and well-deserved award. And I'd also like to thank John Dirks and the Gardner Foundation to have the opportunity to help put this great series together. And of course, the 8 fantastic speakers for agreeing to come out here and enlighten us. I think there's 9 talks, and if this was a baseball team, I think it'd be the Babe Ruth Yankees, except I'd be kind of like Forrest Gump. I'm not sure what I'm doing here, but I'm actually in a position following up on 8 guys and women who have nothing to prove, and I actually have a lot to prove. So I think the tone of my talk is going to be quite different. I've been in Toronto now for 5 years, and my goal has been to come over from industry at Genentech, where I was for about 10 years, and try to kind of implement some of the lessons I learned in industry in an academic environment, which is, can we take basic research and start turning it into, at least, if not therapeutics, therapeutic leads or validation tools that help us define that. And it's been a great 5 years. This is a great community here in Toronto. And what I'm going to talk about is from antibodies to synthetic proteins, which is, okay, hold on. Yep, okay. I'm not sure this is the right talk. Yes, we're not in Shanghai. There we go. It would have been almost the same, except it would have been in Chinese. So, I reside at the Donnelly Center. I just wanted to put a plug into this because actually, this center changed my life. It actually recruited me back to Toronto, which I'm a Canadian, and it's great to be here. And it's kind of the vision of Brenda Andrews, our director, along with all the other faculty that I really appreciated. This collaborative idea of doing big science, systems biology, large approaches, but in kind of a team environment. So there's 30-some faculty. I arrived in 2008, and it was amazingly quick to get started on some pretty ambitious ideas. And I swear I made this slide before Greg Winter mentioned it, and I thought I was being clever, but of course, Sidney Brenner's always more clever than you'll ever be. And the idea is DNA to RNA to protein has clearly been extended in biotech to the idea of drugs. I'm not brave enough to say money, but it amounts to the same idea. If you can develop drugs, you can improve life, you have more money for research. And the change has been over the last 2 decades, the idea of going from single proteins and DNA to omics. And so the central Dogma-omics has become genomics, transcriptomics, proteomics. But the question is drug-omics here? And I think that was one of the promises of large-scale genomics research. The idea was, if we do all of this, we'll learn a lot about the cell, and that's been undeniable. I think we've learned far more than we ever thought we would, but the paradox remains that drugs are still a single entity. It's a heroic effort, and really there hasn't been a major breakthrough in the number of drugs that are approved. And I think a central question for the upcoming decade is to figure out why this is. Is there a systematic reason? And many ideas have been proposed, but I'd like to propose one I think I have in common with quite a few researchers, and that's the idea that although the genome and the proteome and eventually the interactome can tell us a lot about cells on the protein-protein interactions that control signaling, you can't really start making therapeutics until you introduce what I call the antiome. Jim calls this the antibiome. We're going to eventually agree on one or the other. But the whole idea is drugs are simply well-dressed affinity reagents. every drug starts out as an affinity reagent, whether it's a small molecule or a biologic. Really, really good affinity reagents that do exactly what you want become drugs. But it stands to reason then, or I would like to argue, that if you're going to develop more drugs, you need more affinity reagents. Because it's only with tools that intercept and detect proteins at the protein level rather than this level that you can actually figure out what proteins do. If you can't modulate protein function, you can only infer function. And most importantly, even if you understand what a protein does, until you have a molecule you can put into a cell, a mouse, or a person, you really can't alter the fate of that protein. So our goal then has become to convince that we need yet another ohm, and hopefully this will be the one that kind of opens up the promise of large-scale therapeutic development. And we like to do this in a very collaborative, open environment. And that's the other reason for kind of getting back into academics. I think a lot of the kind of stuff we do, we hope will have impact on industry, but it's much easier to do it in an academic environment where we can interact closely. And so I think this has all been explained by much better people than me, but the main problem with an antiome is, of course, we don't really have the technology yet to do hundreds of thousands of antibodies at scale in a renewable form cheaply and efficiently. I would argue the mouse has been a major workhorse for much of cell biology, but when you think of scaling this up thousands of fold, it becomes very difficult to imagine doing that in a scientific manner. So from pioneering work from many people in this room and others, I won't belabor the point of phage display, it does give us this tool to make large libraries, in vitro selections, and kind of get away from the natural immune system. And what's been really powerful here is not only can we now manipulate natural antibodies much, much better, but the theme of my talk will be, I think, and this is the goal of my lab basically, is to progress from natural antibodies to synthetic antibodies, further into other synthetic proteins, and eventually synthesizable proteins, which is one of the holy grails of protein chemistry. And I think not only can we do this, but I think we need to do this if we're going to do the kind of scale up many of us would like to pursue. Proteome level exploration of protein function, I believe very strongly will require at least this to get away from natural limits and probably some combination of these methods so that we can engineer proteins at will that can then be distributed, used in cell assays, used for structural biology, and eventually perhaps be developed into new drugs. So I'm going to have 3 examples that kind of show what we're doing in this area, and also 3 design philosophies in the form of quotes. Since I'm, like I said, I have kind of a interest here to get the point across of what my lab's doing. And going from natural antibodies to synthetic antibodies, back at Genentech almost 15 years ago now, where this work originally started, we were asking the question, not only can we make antibodies, but can we make them as simple as possible? And you know, we quote Einstein here because you can argue with me, but you can't argue with Einstein. Everything should be made as simple as possible, but no simpler. And what that meant to us as protein engineers is to enable understanding and engineering, synthetic antibodies should be made simple without compromising function. So our goal was to make the binding sites as simple as possible, yet retain the specificity and affinity of natural antibodies. And others, including Andreas Pluxton with Morphosis, produced very elegant work, and there's a lot in this area. But I would say we took it even a step further in that we asked the question, if a single framework, a limited set of CDR residues, and only 2 of the most abundant amino acids and binding sites, could we make antibodies that actually work? And what was quite surprising at the time, it's about 10 years ago and still is that libraries that are very, very simple, this is the Herceptin framework, only alterations in length here and these positions, you can get antibodies against many different proteins, some that are very therapeutically relevant, insulin, VEGF, death receptor 5. And in some cases, the affinities of these got into the low nanomolar. This death receptor 5 antibody was 20 nanomolar, could distinguish between human and mouse, could distinguish between close homologs, and yet the structure, just the CDRH3 shown here, was extremely simple. Most of the binding was mediated by tyrosines, yet further work in cells with an anti-VEGF antibody showed these antibodies could be as functional as kind of the gold standard Avastin, which is an approved drug. So despite being synthetic, very simple, these antibodies could function in cell-based assays, and then Germaine Fu went on further with other variants she made to show they work very well in animal models. So this kind of gave us the confidence to say, yeah, this needs to be scaled up and can be scaled up to a level where these can be used in anything that natural antibodies can be used in, and I would say even go beyond that. And the other advantage of having a simple system like this is now you can add additional diversity and understand the consequences. If you perturb a simple system, you can ask whether the perturbation is better or worse. And we made literally dozens of libraries in iteration. Original work was done by Fred Fallouse, but then it's been carried on by people in my lab and other members at Genentech. And what was powerful is we could kind of bootstrap our way from, this would be the equivalent of a Model T Ford to what's now a Ferrari. Just subtle changes. The chassis is not different fundamentally, but little incremental changes have led to libraries. This is our best or workhorse library in Toronto. Jim mentioned a bit about this. And the point here is it's very precisely engineered. I won't get into the details, but every residue is treated as a building block. Some are highly diversified, some are fixed. You'll have length diversity. And the key is this is not random. Each of these changes has been verified and tested as an improvement to the basic scaffold. And by doing this at Genentech and now at Toronto, we and our collaborators have shown that a single library can yield binders that are as tight or tighter than natural antibodies. And the key is structural epitope space seems to be unlimited. As long as you have a structured folded epitope, we can target VEGF, cell surface receptors, membrane proteins, even RNA, and post-translational modifications in ubiquitin. As I said, these were done in collaboration, and much of this was done by Tony Kozikoff and Shohei Koide, working with Joe Piccirilli and Eduardo Perozzo on membrane protein and RNA, respectively. This was done in my lab, and finally this was done by Vishva Dixit and Bob Kelly. But the point is, it was all done with the same library. And we were able to show that these libraries then are highly functional. And since then, Jim alluded to, and I will allude to also, this has become turning the crank. We strongly believe and have shown with hundreds of antigens now, given a milligram of antigen, we can get an endless supply of many low nanomolar antibodies. And I think that will be a game changer for the automation process. And the other thing you can then do is, if you can target any epitope and you can do controlled selections, working with Jason Moffitt on, in the Donnelly Center, we've come up with ideas, and others are pursuing, going after more complex antigens, and the ultimate would be cell-based selections. This has been done for years, but the difficulty has been in decoding what binds to what you want to bind. I won't go into the details, but we've incorporated Illumina deep sequencing methods to analyze the results of phage selections on negative or positive cells of interest. And because these libraries are precisely designed, we can actually resynthesize any antibody of interest. So this means even rare clones can be recovered. So this has saved tremendously on work. And as one example from my lab and Jason's lab by Rashida Williams was targeting this stem cell marker, CD133. It's a 5-pass membrane protein. And the goal here was to develop very high affinity, high specificity antibodies without ever purifying the antigen or even performing any screening. So simply by throwing antibody libraries on cells, doing Illumina sequencing, and recovering key clones, we could say we could get Fabs and eventually IgGs that are very, very selective for CD133 structured form. And they are very good, in fact. They're better, at least 100-fold better than the best commercial antibody for this antigen. And they also, one of them works in Western blots. So this is kind of a new tool we've added to the repertoire that I think allows us to go beyond the limits of even needing a purified protein. And we're working with a lot of groups to apply this to interesting proteins. And kind of the second goal of my lab has been to kind of systematize these methods and make them available to more researchers. I think that's a fundamental goal of any technology. And we've already established what we call the TRAC, or Toronto Recombinant Antibody Center. It's in a separate building from the Donnelly at the Best building, named after Charles Best. And it's fully equipped for developing human antibodies for research, and we hope beyond. You know, we would like to get into therapeutics and diagnostics. And it enables collaborative research. And I like to say it has all of the features of a center except the funding. So this is all supported by grants, but it's been great. I think U of T has been very supportive, providing space, and a lot of people have kind of collaboratively put this together. So our motto is basically, you have to pay to play. You get access to the technology, But if you have that, we can basically do anything of interest. So it opens up the field to many researchers. And I just last count, we had 30 different funding sources, all the way from small to big, supporting more than 50 collaborative projects. So it's kind of a bootstrap method, but I think it's working very effectively. And going further than that, as Jim noted, I think the key is what we've learned from the yeast community and what we've learned from Rosetta, which David Baker pioneered, is things get more powerful when you have a network. So we've already started the recombinant antibody network, which is my group, Jim Wells and others at UCSF, Tony Kozikov and Shohei Koide at Chicago. And this is linking three, I think, pretty good labs, but making them much better because we can work together and solve bigger problems than I think any single lab could solve. And this is coalesced around a large NIH team grant to develop antibodies against all 1,000 human transcription factors, which is we started that a couple years ago, and I think it's progressing well. And it's also allowed us to develop the technology much, much faster, because different groups contribute libraries and ideas, and I think we're well on our way to achieving 1,000 antigens in 5 years, which I think you don't need a calculator to figure it out. I think the genome is then within reach. It's only a scale up of an order of magnitude. So that's been going very well, and I said the next goal then has been, can we use other proteins for specialized purposes? Antibodies are great, they've been fantastic, but there are fundamental limits, and the power of a synthetic approach is any scaffold that can fold in E. coli can be used. And we've been asking the question then, what can we do beyond antibodies for specialized tasks? And the second quote, since I can't top Einstein, actually comes from James Brown, who said, you got to use what you got to get what you want. And what that means for us is, if you look at it, every natural protein-protein interaction is an inhibitor waiting to be engineered. You simply have to make it tighter, more specific, et cetera. And the protein database in particular is a gold mine for rational combinatorial design, which I think we've underutilized. The Rosetta community has done a fantastic job at de novo design, but I think even in natural protein, there are thousands of inhibitors that can be made very quickly. And I'll give two examples of where we've done that kind of approach. One is another approach to targeting P-tyrosine, which Jim gave a fantastic example of de novo design. But you can also look at it as a means of using natural binders, SH2 domains that bind tyrosine. And simply, what we wanted to do here was get enhanced affinity, because these are weak, but maintain specificity. Because our goal would be to develop tools that have the same specificity of, say, the Fin SH2 domain, but rather than micromolar or low nanomolar. And and this is where the rational combinatorial design comes in, it stands to reason to only randomize the region that contacts the tyrosine. And sure enough, working with Sean Lee, we could show with protein arrays and later with cell biology that we get a close to 1,000-fold enhancement in affinity, but the specificity of the protein remains virtually identical to the wild type. So working with Sean Lee's lab, we were able to show that if you introduce these by transfection of DNA into cells as alone, they act as very super dominant negatives. They inhibit the signaling pathways and actually kill cancer cells that depend on Fin signaling. And with the late Tony Poston's lab, Greg Findlay was able to show if you introduce them into cells in the context of a full-length protein, in this case in GRB2 SH2 domain, they act as dominant positives. So because they're tight and they're in the full-length protein, they can constitutively turn on signaling. In this case, it could drive stem cell differentiation in various directions. So, and the nice thing is because this region is rather conserved, we were able to show, not in all cases, but in many SH2 domains, we had a triple mutant that was graftable. We originally isolated it in Fin, we transferred it to SARC, got the same affinity enhancement, and then Tony and Greg transferred it into GRB2 and got the same affinity enhancement. So we think with a couple of these mutations, you can start controlling a large area of phosphosignaling. The other area which we've actually expanded into a lot is looking at ubiquitin as a scaffold, because it's strongly attracting attention as therapeutic targets. Everybody believes, based on RNAi and other methods, mutational data, that DUBs and ligases, and thus ubiquitin missignaling, is behind or at least contributing to a lot of diseases, including cancer. But the problem has been, unlike the kinase field, there are very few specific inhibitors of any of these enzymes. There's a few inhibitors of DUBs, a few of ligases, but it's daunting in that there's 600 or so of these, at least 100 DUBs, and thousands of protein interaction domains. But what they all have in common, and there's great structural data showing this, they all have weak binding sites for ubiquitin. So the idea here is, if they already bind ubiquitin, why not use it against itself? Ubiquitin is, as a dream to engineer, much easier than antibodies, actually. It's very small, it's stable, large binding surface, weak interactions, and critically, it's probably the best evolved protein for expression in cells. So unlike antibodies, it's a perfect intracellular scaffold. So we took ubiquitin, looked at structures of it bound to various dubs, identified a large surface that interacts with these dubs, mutated this, again, with synthetic approaches, you can tailor the library. So here, rather than naive, we have something that already binds. We simply want to make it tighter. We had a very large library, about 7 times 10 to the 10th, but it was mutated in such a way that each member only had 5 to 8 mutations, so a soft randomization approach. So what you get is a highly functional library of ubiquitin-like proteins, about 10 billion of them. And if we expose that library with phage display to any natural enzyme that is recognizes ubiquitin, we could very rapidly get not only binders, but binders that precisely docked at the natural substrate binding site. Not surprising, this is where ubiquitin binds. But it's a great feature because rather, even with antibodies, if you get binders, which ones are inhibitors is often a big question. You hit all the sites. So here, we've shown with many examples, you target natural functional sites. So these are 3 USPs bound to 3 different ubiquitin variants. And as you would expect, in each case, the ubiquitin variant is a very potent competitive inhibitor of the enzyme, all the way down to 2 nanomolar. And none of them inhibits, this is only a few examples, but a large panel of other USPs. So not only are they tight, but they're also very specific. And in these 2 cases, we could then transfect the DNA and show that USP8, which protects EGF receptor from degradation, inhibiting it leads to abrogation of EGF receptor through endocytosis and lysosomal degradation. Conversely, USP21 downregulates NF-kappa B by deubiquitinating RIP. And if we add an inhibitor of that, we get full restoration of NF-kappa B. And now we have many examples of not only developing binders, but being able to introduce these into cells and explore inhibition at the protein level. And we've already developed binders to close to 100 proteins. As you might guess, the bottleneck is using these. So I think we can turn this into a very general method, but already with collaborators at the SGC and Frank Sakari on the structural side and Dan DeRoche and Mike Tyers on biology, these are 8 different structures, ubiquitin variants shown in blue in each case, recognizing very tightly and specifically various proteins. For the 3 USPs, this is another structural class of DUB. This is a E2 ligase. This is an E3 ligase. This is an F-box skip complex. And this is actually a tandem tutor domain. Some of these were known to be natural sites, and some I think we're discovering. And in every case, we're seeing that these have cellular function. They have phenotypes because you're hitting natural protein interaction sites. And you also get surprises, which RNAi, being a knockdown approach, can't afford. In the case of the NED4 ligase, to our surprise, we found one of our binders was a very strong activator, both in vitro and in vivo. So you're seeing hyperubiquitination of the substrate yin-yang-1 transcription factor. So this opens up the opportunity not only to inhibit enzymes, but perhaps turn them on. And if you think about it, some of our most successful drugs, insulin, growth hormone, EPO, are agonists. I mean, we've become obsessed with inhibition of function, but I think there are tremendous opportunities in modulating enzymes in a positive manner. And in other cases, E2 ligases are known to have 2 binding sites. The active site is involved in initiation of the chain, but at least some of these have an exosite that's responsible for chain elongation. And we've shown that we can inhibit either of these, and sure enough, we see different effects. If we inhibit up here, there's a complete blockade of the system. If we inhibit down here, we push it towards shorter chains and monoubiquitination. And again, with RNAi, obviously these subtle changes are not possible. And again, many drugs can be developed against sites which aren't the main enzymatic sites. And the ability to modulate rather than totally kill a protein can often be very therapeutically beneficial. So I hope I've showed you, we think this is a general, well, we've shown it's a general strategy for modulation of the ubiquitin system. Inhibitors, activators, and attenuators are all possible. And an area we haven't explored is perhaps these could also be orthogonal substrates to understand which enzymes work on what. So then finally, to get to the last step, from synthetic proteins to synthesizable proteins, my third quote is from Richard Feynman, of course. What I can't create, I do not understand. And this is, again, a fundamental idea in protein science, which I think has been very powerful. Not only do you want to make proteins that do something, but you want to make them as understandable as possible. In the extreme case, like the Baker lab, where you can actually design these, We are not at that level with everything, but I think to progress, we need to think in those directions. And one thing we had in mind was let's start making proteins that are chemically accessible. This would enhance our understanding of protein structure and function. You can do a lot more experiments with non-natural diversity than you can do with the 20 natural amino acids. And for therapeutics, the ability to introduce a much wider palette of functionality should enhance the activity of proteins. You can make them tighter, more specific, perhaps reactive. So for all these reasons, we wanted to investigate this. And the system we wanted to do it in was to look at small proteins such as this, which our goal was to have antibody-like affinity, specificity, and stability, to do it in D amino acid space. As I'll show, there's evidence that because it's D space, this is metabolically more stable. Natural proteases don't recognize it. And the question was, could it be non-immunogenic? If the MHC complex doesn't display it, because that's chiral, then can you get protein-like molecules that are immunogenically silent, or at least low immunogenicity, and synthesizable, so we can start doing interesting chemistry and apply that on a large scale. And to do this, we actually used an approach pioneered by Peter Kim almost 20 years ago. And I'd like to say this approach was so visionary, it took 20 years for the chemistry to catch up to it. What Peter's group was able to show with SH3 domains and peptides, that what I'm going to show you is entirely feasible. The limits were actually, I think, on chemical synthesis. But since then, my collaborator Steve Kent and others have pushed the envelope on synthesis, where at the milligram scale, we can synthesize, or they can synthesize, things up to 300 or 400 amino acids. And at production scale, proteins of about 50 amino acids, I think, are now generally thought to be accessible, even at drug levels. So our idea was to first use VEGF as a paradigm. This is one paradigm for antibody therapies. Can we get a synthesized D protein that has efficacy similar to an antibody? So Steve's postdoc, Kalyan, synthesized VEGF-A, the natural protein. And what you see, these are real crystal structures. It looks exactly like the natural hormone. But the nice thing is you can do the same synthesis virtually in D space. So this is D VEGF, its crystal structure, and there's a mirror plane. I didn't do this with Photoshop. This is actually, these are real structures I'll show you. And then this was handed over to my postdoc, Marudi Upalapati, who took a library of small scaffolds based on GB1 and identified binders in L space that recognize D VEGF. And here's the structure of D VEGF, completely unnatural protein, bound to an L VEGF. And again, these were made by synthesis. And then finally, the trick that Peter's group pioneered was, if you can do this by selection, you can do this by theory. In other words, if you now make the binder in D space, exact same sequence, it will bind with the same affinity and specificity to the natural L protein. So the key here is you don't have to display D proteins to get D binders, as long as you can synthesize D antigens or work with somebody who can synthesize D antigens. So this whole cycle we went through, and in fact, here's the mega structure that was solved. This is the largest synthetic structure we know of. So all of these proteins were chemically synthesized. You see L-VEGF dimer, D-VEGF dimer bound to 2 D inhibitors or 2 L inhibitors. And the crucial area of this structure for us was, even though this is an unnatural D protein, it very precisely mimics the epitope of VEGF binding to VEGF receptor 1, VEGF receptor 2, or the therapeutic antibody Avastin. So we predict that this should act as an inhibitor. Sure enough, we're advancing along that steps. The original binder was rather weak and not that stable, but through 3 rounds of optimization of both stability and affinity, we were able to get affinity down to 1 nanomolar, which is very comparable to therapeutic Fabs, and we were able to get TMs down to greater than 95 degrees. So you can't even denature this unless you add urea and boil it. And the nice thing is, it does prove to be, at least in preliminary experiments, lower immunogenicity than the L isomer. So we think we're on the right track. And in endothelial assays, it downregulates VEGF receptor signaling. This is down to basal levels. And now we're looking at proliferation assays and moving this into animal eye models. So we think it's quite encouraging. And going beyond that, since you can use anything that's small, we've decided to expand diversity space by exploring many shapes. Marudi's put 70 scaffolds on Phage, and we're in the process of making libraries based on all of these. So I hope I showed you with these kind of philosophies that we are progressing towards kind of a goal of developing the tools that we can then apply on the genome scale. I'd like to thank my group, Helena, Andreas, and Marudi, and also Fred Fallouse, who did the original antibody work at Genentech, Pankhead Way and Marina are continuing with Ubiquitin. Great technical support. Haiming did the Superbinder work, and a lot of collaborators everywhere. Thank you.